Thank you, Serban. And I think it's appropriate pronouncing my name like that, given that I'm in Serbia. So at least for once in a context, it sounds like it's an interesting name. So listening to the talks today, I was like realizing, oh, I have nothing to say about policy. I'm just coming with this sort of old questions about epistemology of modeling and talking about Turing models and their relationship with experiment. For me, is just an excuse again to try to say something about model-based explanation. But it's also saying something a bit about a clash between two research cultures in biology. Kind of, let's say, theoretical physics style of work, mathematical work in biology, and experimental work in biology. And I want to make a case that the historical discussion that Turing started in 1952 about how to understand biological patterns is a nice case study to focus on to tackle some of these questions about how biologists might want to get an explanation of biological phenomenon using other tools that then they were used to. So in part, this is kind of half historical, so I'm going to give you some details about these Turing models. I'm going to assume you're not very familiar with them. Turing is sort of known for his kind of contribution to computer science and artificial intelligence, and I hope that at least it will be a piece of news for you that he was also very important in developmental biology. I mean, biologists haven't liked kind of to admit to this until very recently. So at the centenary in 2012, all of a sudden, like Turing was like a pioneering figure for developmental biology. This was not a fact that was historically acknowledged for a long time. And I'll say a bit about that and why I think this is the case. But also kind of move into more recent research and kind of show how mathematical modeling of this kind is linked to experimental work. And why do I insist on this kind? Because nowadays, mathematical modeling in biology is mainly driven by data. We have lots of experimental data, so it's a lot of effort invested into devising analysis methods of the data we have. Unlike these cases, the Turing type of modeling, I will kind of talk about it in different ways. I'm going to talk about it as fictional modeling, as minimal modeling. It's something kind of sounds strange to the ears of an experimental biologist. It's kind of seriously like biology done from the like mathematician's armchair, and see how that works and what's special about it. Okay? So I'm coming this case study with two questions I'm trying to bring together. They come from different parts of my research, as it were. So one question is, when we have a really false model, like one like that's so false, should we still trust the type of inferences we draw from that model about the target system? So when it's really false. So when I say fictional, I mean like models that are so false. I'm not going to say that much for those of you who are a bit more familiar with those types of philosophical discussions about the ontology of models here. So I'm just kind of picking up on an idea Peter Godfrey Spitz put forward, I think initially, that the fictional character of models is something distinctive about model-based science. And more serious kind of philosophical discussion started from there. Maybe we can learn something about the ontology of models, how they differ from experiments in that way. But I'm not going to go into those debates. But rather, I want to use the term to kind of pick a particular class of models that make many falsifying assumptions. And I'll say more about that. The other question, I'm actually kind of, this is a piece of biography more than anything. I found out myself about the Turing models of morphogenesis because I was looking for an example of a computer simulation that I can follow, like from beginning to end. So I was like really just looking for a manual, like performed computer simulation. And Turing was the first manual performed computer simulation done in kind of empirical science. So I was like, okay, I was trying to understand how the simulation works there and what are the features of the simulation, and then I stumbled upon interesting questions, so I'm trying to bring them together. And so I started to think about what can Turing model or this case study show us about the difference between modeling and experimenting. Okay, so I hope to say something about that. So the plan would be, say, I'll introduce you a bit to Turing patterns. introduce the broader and situate it in the broader problem of modeling and experimenting, whether we can really distinguish between these two scientific activities. And then in order to kind of ground better my conclusions about what are the epistemic outputs of doing this type of modeling, I'm going to look at two more recent applications of Turing models, and then kind of propose this thesis that these really false models can explain something in developmental biology, okay? Now, Turing, in the 1952 paper, started with a very general and very ambitious question, and in a way, a question that was fundamental to developmental biology, but that no experimental biologist really wanted to ask because it was too complex to admit to kind of a well-formed type of answer. And the question was something like, if all the cells of the organism have the same genes and basically kind of the same type of enzymes, how is one to account for the development and organization of so many different kinds of cells that are required for the characteristic structures and form of a complex organism? Okay? So that was a really big question. How do you get the variety of form you observe in a multicellular organism from cells that are basically the same? And his proposal is what I think in the literature is actually the first time when the term was coined, the first mathematical model that's based on a system of partial differential equations that characterizes the reaction and diffusion of two substances. And I'm going to say how Turing got from the first general problem to his model via which type of assumptions, then how he analyzed his model, and what were the inferences drawn from that model. But basically, from the beginning of the paper to the end of the paper, he makes the following claim. The minimal model that I'm proposing is capturing relevant biological features, irrespective of all the idealizations and all the simplifications he introduces. Okay? Another important point, Turing was not drawing on any experimental data whatsoever. And he doesn't make a claim in the paper that experimental data is necessary to validate his argument and the sort of claim he makes based on his model. Okay, and I think these are important and distinctive features. So what are the assumptions Turing makes? So first, as developmental biologists recognizing, you have the complexity of cell structure. So how do you get from that mess to something that you can model and describe mathematically? So you can go for a discrete type of approach and treat cells as points on a lattice, or you can just treat the whole embryo as a continuous structure. Now, if you choose one or the other, it doesn't really matter. In both cases, you can define the state of the embryo by a state equation. Initially, Turing said, we're going to need mechanical variables to show how you get the physical transformation from the kind of embryo to these differentiated cells. And you'll have mechanical variables, and they're governed by Newtonian laws of motion. And he said you'll have chemical variables, concentration, the rates of diffusion, that are governed by the reaction kinetic equations defined from the law of mass diffusion. But then, because so little was known about the interaction between the mechanical variables and the chemical variables, he said, okay, we'll just leave to aside the mechanical variables altogether and just consider our system as being the sort of two interacting chemical substances, and we are just going to focus on the chemical dynamics of the non-growing tissue. So remember, we started with a cell that was growing, differentiating, and supposed to kind of get us all this diversity of form, and all of a sudden, we gave up on the non-growing, on the growing, so it's a non-growing. It's just a system of chemical substances, and assumed that the mechanical forces would somehow build on the chemical gradients of concentrations to shape the actual physical system. Okay? So again, this is the kind of simplified picture how you get the assumptions. You start with this general question: how do embryonic cells differentiate into different cell types that form the tissue for different organs? Via those assumptions I mentioned, you get to this very simple model that chemical gradients through this embryo could give rise to spatial heterogeneity, which then would kind of influence gene transcription and drive cells to differentiate organs in the body. So that was the very simple picture he was proposing, and the thing in the middle was the core of his mathematical model. Okay? Now, say we are going to focus, this model is focusing on chemical gradients through the embryos that give rise to spatial heterogeneity. So it's really important that spatial. Now, there is a question how they might be set up. And Turing's proposal was they are set up via diffusion of chemically interacting substances over some spatial domain. So these are kind of the key elements of his proposal. And you can model that. So if you take these two kind of interacting substances, you can model that via a set of like diffusion, reaction diffusion equations. And you can then, these are the definitions, and then you can analyze the system and try to see what happens when you impose certain conditions on the behavior of the two substances. And that analysis is going to give you, and in this case, gives Turing some interesting results. And in terms of definitions, is important. I mean, this is kind of should be at the end, but I thought it's a good idea to put it at the beginning. In the terms of this model, a pattern is the stable, time-independent, and spatially heterogeneous solutions to those equations. So that's what the pattern is in the model. So I think I want to stress this claim because sometimes it's, okay, so what does the model derive? So what kind of derives these types of patterns? So when you're going to say that the model, it kind of, what's the target? Are these patterns that are understood mathematically in this way, okay? How are these patterns generated? Via so-called diffusion-driven instabilities or Turing instabilities that occur when a steady state, which is steady in the absence of diffusion, becomes unstable when the chemical substance is diffused. Okay? The mathematical model I just showed is analyzed. If you first determine the conditions for instability, so what has to be the case mathematically to have stability in the absence of diffusion, and what it takes to have instability with diffusion, like small perturbations in the variables that you choose. And then, can solve this, and the result of the manual computer simulation performed by Turing showed that there is a special sensitivity to the spatial domains of the solutions. So let's see whether this is kind of the visualization of the simulation for like running it for just a system with a single substance. And in that case, actually, you would get kind of a stripe pattern in a single dimension. Here, it would be kind of a more salient pattern in a single dimension. Now, if you move to 2 dimensions, you'll see that if you kind of have a very small width, you'll get a pattern. So from the reaction diffusion equations, you will get a pattern that looks like a stripy pattern. Now, when you are kind of in a square, so you have the wider width, you would get kind of instabilities on both axes, and you will get a pattern that looks more like spots. Now, Turing, after doing the analysis, he said this is a good model of how patterns are formed or how kind of cells differentiate in development. And basically, what was his justification? He said there is a resemblance between the patterns we see in nature and the solutions to my equations. So that was the justification. It was a crude resemblance between the shape of the solutions and the patterns observed in nature. And that was the extent of it. Now, I want to make a point that Turing, at least in the original paper doesn't make, but James Murray and others commenting on the relevance of the Turing modeling technique for developmental biology did point out. Namely that despite all the falsifications involved, postulating diffusion and reaction as the main processes that drive biological pattern formation was compatible with biological knowledge at the time when Turing was writing this. So the fact that Turing ignored genes in 1952, the fact that he assumed that the inhomogeneities that you find in the cytoplasm of the cell don't make a difference to biological pattern formation, were biological assumptions taken for granted at the time. So in a sense, what he proposed, namely biological pattern formation, being the result of a diffusion-driven instability in a system of reacting chemicals, was, if you want, the only physically plausible mechanism at the time. So relative to biological knowledge, all those falsifying assumptions that he introduced to get from the very complex embryo to his mathematical equations, and by the way, the kinetics in those equations is imaginary. He was not, as I mentioned, he was not drawing at all on experimental data and how the kinetics might look. But in virtue of the fact that there was this compatibility with the key assumptions about biological development, I think a stronger claim can be made and a bit kind of improved on the justification of why this might be a biologically adequate model. Okay? Nevertheless, this model was not well received by the biological community. I mean, basically it was ignored. And I think the question of biological relevance was the one that haunted that type of modeling strategy. Because Turing was not making an effort to relate it to experimental data. And he was kind of characterizing as a mathematical modeling in biology, also a bit, this is kind of a separate commentary, but relating to a tradition that was not very well received at the time. So for instance, let's say at the, what was it? So it's kind of a very important symposium on quantitative biology in 1934. Charles Davenport said this about, I don't know if you can see, but I will just read it. In a general report assessing the state of the field, this is kind of biology as a large, and reacting to kind of the increasing value of kind of bringing mathematical tools into biology. He said, it's futile to conjure up in the imagination a system of differential equation for the purpose of accounting for facts, which are not only very complex, but largely unknown. It is said that if one asks the right question of nature, she will always give you an answer. But if your question is not sufficiently specific, you can scarcely expect her to waste her time on you. What we require at the present time is more measurement and less theory. And I am exaggerating a bit, but that still seems to be the ethos in many areas of biology. More experiment and kind of less theory. So there seems to be kind of an implicit assumption that there is a striking difference between modeling and experimenting. And that experimenting, at least when it comes to biological systems, it gives you sort of epistemic privileged access to the phenomena you are interested to explain. Now, this is a topic that has been discussed extensively in the philosophical literature, and different authors have proposed different criteria for distinguishing between the two types of activities. The reason I left the table just empty is because I don't think the verdict is still out, whether any of these criteria separately or as a cluster can actually distinguish between the two. And also, I kind of cut out the separating line because it seems to be more agreed that depending on the case, the continuity between the modeling and the experimenting this kind of has different degrees. Typically, the question of the material character of models and the fictional of experiments, and the fictional character of models seems to kind of tear them apart. But that's always kind of put in the context of what's the relationship between the model and the target. So I just said in the case of Turing that many, or I mean I suggested if I not said it explicitly, many of the idealization, falsifying assumptions he made were like driven by tractability, getting to some equations that you can then analyze. They were not driven by specific facts or hypotheses about what's happening during biological development. Okay? And then, I mean, there are, on the other dimensions we can discuss if you think one of them is particularly promising. But I think there are strong cases for thinking that it's not a clear-cut distinction between the two. And maybe that's fair enough, but there is a lingering question. And I think kind of a biased answer to this question that kind of derives from this attempt to distinguish between the two. Namely, concerning the quality of the inferences made based on inferences versus the quality of the inferences based on models. So are the inferences made about real target systems more direct and more reliable if we base them on experiments than if we base them on mathematical modeling? In a way, you might say, sure, you just, the only example that we discussed so far in this talk is a kind of a model that, calling it as a resemblance to reality is already a stretch of the imagination. So, of course, it seems that if I engage with the kind of, by experiment with developing embryo, that's going to give us better inferences and conclusions to draw about it. But actually, I think that that's not always the case. So I want to, before getting to my kind of what's my stance on the relationship between two, here are zebras again, present a kind of an application of Turing models that have incorporated some kind of experimental results in order to kind of provide basically the same type of explanation that Turing was getting at. So the question that James Murray's study in How the Leopard, I know those are not leopards, but got his spots, was how does the same process give rise to patterns in different organisms? Why do I say the same process? Because the molecules involved in kind of generating pigmentation on mammalian coat patterns are pretty much the same. They are very similar. So in a sense, you might say if I'm going to kind of look at the proteins that are involved, they are the same. Now, why do I get spots in some mammals, stripes in others, things like that? What Murray did, he ran a simulation on a similar kind of reaction diffusion system. Now, these equations included only, if you want, like, the type of information that you saw was presented, it typically including in the reaction diffusions, namely no specific information about the proteins that were involved. The analysis of the results pointed to the same type of spatial sensitivity of the patterns. So the biological processes involved are similar, the patterns differ, and what the model shows is that, if you want to use a term that's really popular now in philosophy of modeling, the difference-making factor here and the explanatory factor is the sensitivity to kind of this spatial sensitivity. It is not identifying the particular proteins that are involved in these processes. Okay? In a way, a similar type of application has been kind of performed several times by Hans Meinhardt, where he doesn't look at how the same process gives rise to different patterns, but rather how different processes give rise to similar patterns. Why different processes? Because, again, the proteins involved in generating different relief and pigmentation patterns in different types of T-shells are different, but you kind of notice the same kind of shape pattern. So, again, the same sort of strategy, the same type of explanations that describe the dispositional properties of the substances involved and their geometrical distribution. And the explanations, the explanation here cites as the main explanatory factor this spatial sensitivity, rather the identity of the material causes involved. Okay? So 3, if you want, general lessons. You might think they are not directly supported from what I said now, but we can kind of discuss that a bit more. I think when you look more broadly at the career of Turing models in developmental biology, you see that there have been explicit efforts to kind of check whether the type of processes postulated by the model can actually give rise to patterns. And, I mean, a striking result was in Pittsburgh in 2014, where they actually created a synthetic system that was governed by the reaction diffusion system with kind of modified kinetics, but very similar to Turing, and they produced the type of patterns, at least the 6 types of patterns that were generating via the Turing simulation. So that was considered a very strong experimental validation of the model. Sure, it was in a synthetic chemical system, it was not in a biological system. Nevertheless, I want to kind of point to that instance of extending the modeling efforts because it shows that there are often instances where modeling and experimental results are triangulated, but also often there remains a gap between them. Now, I think that when you're trying to build up a case as I want about the complementarity of modeling and experimenting, it kind of blocks the epistemic privilege of one activity or the other, that we should do just experiments or more experiments and less mathematical modeling of this kind in biology. And the sort of claim that I still need to unpack more, and that's basically because I'm dissatisfied with the options I've explored so far, is that the good justified inferences based on either on models or an experiment have to rely on the relevant link with the real target. And why do I say relevant link rather than saying some similarity relation or some representation relation? I hope I'll leave that hanging because I want to say something a bit later. Now, if you want a kind of general question, I want to say, I didn't want to kind of set up the talk and saying how do Turing models, what are their benefits? Because I think there are many, and if you look at their applications across developmental biology, I think you can pinpoint several functions that they play. I want, though, to make a claim about their explanatory value, because I think that can be articulated in a way which is more clear than has been done. I claim that they provide, if I say like structural explanations, call it here, biological patterns are explained in terms of structuring causes, namely the conditions under which they are formed via different or similar biological processes. The structuring, what I call here the structuring causes, I think the term originally, at least I know it being introduced by Dresky in 1988, don't describe kind of the type of things that are that figure in kind of efficient causal links. So they are not material causes. They are not specific proteins or genes. They are the conditions that need to be in place for a certain type of process to occur. So that's what I understand by. I take it to be pretty innocuous, the fact that I'm calling them structuring causes, but it's essentially that they are understood as the sort of conditions under which patterns are formed. Now, in terms of what's the accepted landscape of explanations in developmental biology currently, Marco Nathan has a great paper in which he says, oh, we should kind of be more pluralist. We are not just focusing on genetic explanations in developmental biology, although for a long time the focus has been on them. So not only explanation that cite as explanatory factors, proteins, and other types of molecules and their interactions, but morphological explanations as well. And morphological explanations are exactly the type of explanations I pointed to, explanations of biological patterns that don't mention the identity of proteins involved and many of the details that you would really want to have at the molecular level. Okay? And then he points out that actually you can combine the first two classes of explanations and get morphogenetic explanations. And I think the work where Turing models have been used most fruitfully, like with alongside with experimental work, is to generate this type of morphogenetic explanations in early development. So I'm just pointing to the fact that we can understand this class as broadly understood as morphological explanations because they are explanations that just focus on form, as being structural explanations in the sense pointed out. Okay, so in terms of general conclusions, I tried to kind of paint a broad picture in which, yes, there is a difference from the broad explanandum or question with which Turing started about explaining biological morphogenesis and the kind of real targets of the models in relation to which we can kind of make a claim about their explanatory values. So we can make a claim that the explanatory link exists between the model and kind of biological pattern formation. And I linked kind of this way of understanding the type of explanation provided by these models to this kind of classification of explanations provided in developmental biology, and said that morphological explanations are a type of structural explanations. And the part of the story you haven't heard, and that it's still in the paper that's still very long, is that if you are trying to kind of carve out the different ways in which these really false, so false mathematical models are used, you can point out that they can shape and actually direct experimental design. So there are cases, examples where you can point to these functions. And also that the results of mathematical models can be used to the results of these sort of triangulation efforts can be used to update, change, or discard the model. Not discard yet, but I said I'll put it there for the sake of completeness. Thank you.